Happy Friday. Happy Olympics. I think that is starting today. So if you've been watching from the United States or abroad, I hope you guys enjoy it. Okay, we're going to go ahead and get started. My name is Sam Stewart. I'm a project manager here at the Data and Research Center, specifically working with our support core. We're really excited today to talk about the structural variant data and the All of Us Researcher Workbench. So I'm going to introduce our speaker and then pass it on over to her. So Emma Pierce-Hoffman is a computational associate in Mike, I can never say his name, Tolkowski's lab at the Broad Institute of MIT and Harvard. We're really excited to have her here today as a really good structural variant expert and someone who really helped get that structural variant data that we had in our off-cycle release last month up and running. So her work focuses on computational methods for structural variant detection, and she led that generation, like I mentioned, of the SV data for the All of Us Researcher program. So she's going to talk about that today. This is being recorded, so I will post this information on the User Support Hub in the next week. If you have questions, we do kindly ask that you hold them to the end of the presentation, but you're more than welcome to add your questions to the chat, and myself or one of my colleagues will answer it. So Emma, I will stop sharing my screen and pass it on over to you.
Great. Thanks so much for that introduction. And let me get my screen up. All right. Thanks everyone for coming. And I am excited to talk today about the structural variant data in the All of Us research program. So why today? Why are we here? Last month, we actually released new structural variant data and really expanded the data that's available in the controlled tier of the Researcher Workbench from SVs on about 11,000 participants to nearly 98,000. So it's a massive increase. And from all of those participants, we were able to discover about 1.5 million structural variants. So those numbers actually make All of Us one of the largest and most widely available catalogs of SV data. So it's really exciting both for the program and for the field in general, because studying structural variants is essential to understanding genetic diversity across populations, how genomes have evolved and changed over time, and the origins of different health conditions. So we're really hopeful that this new data will provide increased power to do all of those analyses. So I'm going to take a step back and give a little bit of background and make sure that we're all on the same page. First, what are genetic variants? So a genetic variant is a difference in DNA sequence between individuals, or often we're talking about a difference between an individual and the reference genome. So I'm showing an example here in which the reference genome has an A at one position and the example sequence has a C at that position. So that's an example of a single nucleotide variant, or SNV, in which just one nucleotide is altered. But that's not the type of variant that I'll be focusing on for the rest of this presentation. I'll be talking about structural variants, or SVs, and those are larger genetic variants. So those are DNA sequence alterations that affect greater than 50 bases. And I'm showing an example of one type of structural variant, a deletion here. So you can see that this light blue region is present in the reference genome, but it's missing from the sample genome. And the point of divergence between the reference genome and the sample genome is called a breakpoint. And in addition to deletions, there are many classes of structural variants that are shown here. So along the top row, we have copy number variants, or CNVs, and these are rearrangements that involve the gain or loss of a copy of a region of DNA. So that includes deletions, which we already saw, and duplications, where a region of DNA is copied again, and multi-allelic CNVs, where the copy number is variable in the population. Along the bottom row, we see balanced SVs, and these are rearrangements that do not affect the copy number. So those include insertions of novel sequence relative to the reference, inversions in which a segment of DNA has its orientation flipped, and translocations which are rearrangements involving whole parts of chromosomes. Up in the top right, we also have complex structural variants. So these are more complicated rearrangements that have more than one of these other types of SV signatures. And the last type of structural variant that you might see in the All of Us data is break-ends. And this indicates that we found some kind of evidence of a rearrangement, but we weren't able to fully resolve the type. And as I mentioned before, SVs play an important role in human disease. So I'm just showing screenshots from a couple of studies, but there are many more out there that have found SVs to play a role in disease, ranging from congenital skeletal defects to neurological disorders. So this is one of the reasons that SVs are so important to study. Okay, so now that I have defined structural variants and hopefully provided some motivation for why we want to study them, let's talk a little bit about the data that's available in All of Us. So we performed structural variant calling on 97,940 short read whole genomes and discovered more than 1.5 million structural variants. So this figure shows the breakdown of those structural variant sites by SV type. So you can see that deletions have the highest count, followed by insertions and then duplications, and there are smaller counts of the other SV types. We can also look at this on a per genome level. So the median count of structural variants that we discovered on an individual genome is 9,686, and this figure again shows that also broken down by SV type. And we did extensive analyses to ensure the quality of this data set. So I will show a summary of some of those benchmarking statistics here, but I also encourage you to check out the documentation on the User Support Hub that details all of the QC measures that we performed on this call set. But to summarize here, we compared to matched long-read PacBio sequencing data and calculated an 88% confirmation rate. We compared large copy number variants to evidence from microarrays and estimated that there was only about a 3.4% false discovery rate. We looked at Hardy-Weinberg equilibrium, and we found that over 93% of SVs were in Hardy-Weinberg equilibrium, and there was strong linkage disequilibrium between SVs and nearby SNPs and indels. So all of these are really positive signs ensuring that this data set has really high quality. Okay, so I want to very briefly talk about the methods that we used to generate this data because I think it can help to have some background understanding of those methods when you're using the data. So as I mentioned a couple of times, this was done on short read whole genomes as opposed to long reads. And SV calling from short reads is difficult, but it is still really highly informative for research. And we used the GATK SV method, which is the same method that was used to generate SV calls in the Genome Aggregation Database, or GNOMAD. So when I say that it's difficult to call structural variants in short read data, that's because short reads are short. If a read is only about 150 bases long, but a structural variant can be hundreds or thousands or millions of bases long, it's not enough to just look at the divergence between that one read and the reference. So instead, we look for characteristic patterns of 4 different classes of evidence. And those classes are split read, or SR, paired end, or PE, read depth, or RD, and B allele frequency, or BAF. And I don't have time today to go through all of these in detail, but they actually are all visible in IGV. So let's just take a quick tour through what you can see in IGV. This is a real-life example of a heterozygous deletion, and the deleted region is shown by this blue bar up top. And we can see each of the 4 classes of evidence here. So going one by one, first, if you zoom in on either side of the deletion, which we have done here on the right side, you can see these rainbows of mismatches at the end of some of the reads. And those are soft clips or split reads. So this deletion does have split read evidence, and split read evidence actually tells us the exact start and end point of this deletion. You can see right here is where the mismatches start. The next type is paired end or PE evidence. So here we have colored and sorted the reads by their insert size. So these red reads up here, you can see are much farther apart from their mates than the gray reads in the background. So this indicates that there is PE evidence supporting this deletion. Next, we can look at the coverage track. And you can see that there is a dip in coverage across this heterozygous deletion. So there is read depth evidence supporting it as well. Finally, for B allele frequency, we are looking at the SNPs that are present in the deleted region. And you can see the allele fraction of each of those by looking again in the coverage track. And the allele fraction for each of those SNPs is 1, which is what we expect in the presence of a deletion because there's only 1 copy of that deleted region. So putting that all together, you can see that there is strong evidence for this deletion based on those 4 classes of SV signatures in short read data. But we don't perform SV calling by just looking at each structural variant one by one in IGV. So let's talk briefly about the GTK SV pipeline. GTK SV takes an ensemble approach, applying 5 different SV discovery algorithms and utilizing all 4 of those different evidence types that we just talked about. Then we perform variant merging to reduce redundancy, joint genotyping of all sites across all samples. We resolve complex rearrangements, perform filtering to improve specificity, and annotate predicted and functional consequences and allele frequencies. And this pipeline is designed to run in the Google Cloud Platform, and we applied it in the Terra interface to produce this all of us data. Okay, so now that we've talked a little bit about how we produce that SV data, let's take a look at some examples and talk about how to understand and interpret the data. So we produce SV calls in variant call format or VCF. And this is an example SV taken from a study of genetic diagnoses for fetal structural anomalies. And this is just one line of the VCF, but I've broken it up across multiple lines to make it a little bit easier to read. And I've added the corresponding field from the header above each of these fields, again, to just make it a little bit easier to read. So what can we learn from this VCF record? First, just like with a SNP or indel VCF, we can look at the chromosome and position to understand where this variant is. But remember that many structural variants are not just at one position, but actually are over an interval. So we've found the chromosome and we found the start of that interval. But to find the end, we have to look in the info field for the end tag. So once we have the chromosome position and end, we have the full interval. Next, we might want to know what kind of variant it is. So we can look at the alt field or the SV type field to see that it is a deletion. And there's also an SV length field that tells us that this is 1,369 bases long. So we've learned a little bit about what type of variant this is. Next, we might be interested in a particular sample. So For sample X, we see that the genotype is 0/1. So sample X is a heterozygous carrier of this variant. And we can also see the different types of supporting evidence that were used to call this deletion in sample X, and those are RD, PE, and SR. We're probably interested in the quality of this variant as well. So on a site level, we can see that the filter status is pass, and the qual score is 50 out of 99. On a sample level, we can see that the GQ is 49. We also have quality scores for each of the supporting evidence types. In this case, the PE GQ is 89, so there's really high PE support for this deletion. If we want to further assure ourselves of the quality of this variant, we can take a look in IGV. So the deletion region is shown in red here, and we can see, similar to the previous IGV example, that if you look really closely on either side of this deleted region, you can see those rainbows of mismatches that represent soft clips or split reads. You can also see, again, where we have colored the reads by insert size. So the red reads indicate discordant pairs, so there's PE evidence as well. And there is a small dip in read depth across this deletion. So you can see all those different types of supporting evidence again in IGV. Our next question might be, how common is this variant? So this was from a cohort of about 900 samples, and there's only an AC or allele count of 1. So this sample is the only carrier in a heterozygous state, so it's pretty rare. And this was from a clinical study, so we are definitely interested in what is the clinical interpretation of this structural variant. And you can look for the functional consequence annotations in the info field. This variant has the annotation predicted loss of function in the gene MED13L, and that gene is actually associated with a developmental disorder that is consistent with sample X's phenotype. So this is a really strong candidate for the cause of disease for sample X. Okay, so I'm going to skip over to a demo now to show you how to actually work with this data in the All of Us Researcher Workbench. So we have created a Jupyter notebook that you can follow along with either now or later on your own time. And I want to show you first how to find that. So if I go to the homepage of the Researcher Workbench, I'm looking for this workspace called Workshop Intro to All of Us Genomics Data. And it's on my front page because I've clicked on it before. But if it's not for you, you can go to Featured Workspaces, and under Tutorial Workspaces, if we scroll all the way down, it's in the bottom right, Workshop Intro to All of Us Genomics Data. If we click on that and then go to the Analysis tab, we're looking for Exercise 8, Explore Short Read Structural Variant Data. And I have a copy of that notebook already open over here, so that's what I'm going to go through. And I have already run all of the cells in this notebook to produce the outputs to save us a little bit of time. So I might go a little bit fast if you're trying to follow along at the same time, but there's lots and lots of documentation in this notebook, and this session is also recorded, so you can always go back and listen to what I have to say if you're falling behind. Okay, so we want to start by just showing you where to find the structural variant data. And as with the other types of data, you can find the file paths for this data in this controlled CDR directory, which is linked from the notebook. So if I look for structural variants, it gives me all of the paths that I need. So I copied and pasted one of the paths from that website here. So this is the folder for the V7 off-cycle release. That's this latest release of nearly 98,000 genomes with SVs. And I can see that there are 2 subdirectories here. And if you're not familiar with Jupyter Notebooks, I want to just take a moment to show you how to run these cells. So you can click on the cell and hold down Shift and press Return. And while that cell is running, you'll see that there is an asterisk here. And once it stops running, the asterisk disappears and you can see the output. So there's two subdirectories in this structural variant off-cycle release directory. So there's variants in the VCF subdirectory, and there's also auxiliary data. So let's take a look at what's in each of these subdirectories. So we're going to take a look at the VCFs first. So if I, again, run gsutil-ls on that subdirectory, that's going to list all of the objects that are within that subdirectory. And I can see that there are, again, two subdirectories, full and sites only. This is because we release structural variant data in two different types of VCFs. The full VCFs contain genotype information for all of the samples, and the sites only VCF does not contain any genotype information. And the reason that we do that is that without all of that genotype information, the sites only VCF is a lot smaller. So if you are doing an analysis that does not require using any sample genotypes, it's probably going to be easier to work with the smaller sites only VCF. So let's find the paths for the sites only VCF. Again, we can do that by running gsutil-ls on that sites only subdirectory. It's a very long path, so I just scrolled over to see the full path. And when I run ls, I can see that there is the sites only VCF at this path, and it also has a corresponding TabX index. The full VCFs with sample genotype information included are very large, so we've actually split them into one VCF file per chromosome. So when you run gsutil-ls, you'll see that there are 24 of these full structural variant VCFs, and they're labeled with the name of the chromosome in the file name. And each of these VCFs also has a TabX index at this VCF path.tbi. Okay, so now that we've shown you where to find all of the variant calls in VCF format, let's take a look at what's in the auxiliary data. So yet again, we're going to run gsutil-ls on that auxiliary data path, and we see that there are three subdirectories: aneuploidies, known issues, and relatedness. So for known issues, there's only one known issue affecting this release of structural variant data, and that is that there's a small number of participants that have SV calls, but they don't have associated data in the CDR. And this file provides a list of those sample IDs that are affected. And to learn more about this known issue, or really about any aspect of the structural variant data, I've linked to the data quality report. We also saw a subdirectory for relatedness. So if your analysis requires that all the samples be unrelated, we provide a file containing a list of the minimal set of participants that you would need to remove from the SV VCF to retain the maximal set of unrelated samples. So this is that list, the minimal set of related samples to prune from that VCF. And the last type of auxiliary data that we have here is aneuploidies. So we provide lists of samples that have germline aneuploidies of allosomes, so that's chromosome X or Y, mosaic aneuploidies of allosomes, and mosaic aneuploidies of autosomes. So we have three separate files of aneuploidies for each of those different categories of aneuploidies. And we've documented the format of each of those files on this web page that I've linked here, how the genomic data are organized. And we can also take a look at those files right now. So what I'm doing in this cell is I'm just printing out the first line of the mosaic allosome aneuploidies table. which is the header. So we see that there is the research ID, so that's the sample or participant ID, the affected chromosome, the estimated copy ratio for that chromosome, and the type of aneuploidy that we observe. I have also run a command to count the number of lines in that file, and the output of that is 950. So there's 949 of these mosaic allosome aneuploidies in this file, plus 1 line for the header. Okay, so now that we have seen how to find all of the data that's associated with this structural variant release, let's actually take a look at how to use some of that data. So we're going to do a very simple analysis. This is just meant to show you a little bit of how to work with our VCF files and get you started with some code snippets as a jumping off point. So what we're going to do is we're going to extract variants in a gene or gene list of interest. And we're going to demonstrate 2 different ways to do that with a Python script and with BCF tools. And we're also going to take a look at some of the VCF records that we extract and go through them like we did in the slides to understand what they mean. And since we are recording this demonstration, I have limited all of these analyses to variants that have an allele count or AC greater than 40 to ensure that all of the examples that we're showing pertain to at least 20 participants. But if you're going through this on your own time, you're welcome to remove those limits. We just can't show those publicly. Okay, so in order to extract these variants of interest, we're going to start by localizing the sites-only VCF. As I mentioned, the sites-only VCF is a lot smaller, and this analysis doesn't require sample genotypes, so that's why we're using this VCF instead of the full VCF. So this cell defines the sites-only VCF path and its index path from where we found it earlier in the notebook. And before I localize this VCF to the notebook environment, I'm going to check how big it is. I think that's always a good idea. Before you download data, you want to know how big it is if you have enough space. So it turns out that it's only about 1 gig, so it's definitely not too large, and I think it's safe to localize it to the notebook environment. So that's what we're doing in this cell. We are using gsutil cp to copy the sites-only VCF and the index to the notebook environment. And I've already executed this cell, so when I run ls on to list the objects in the notebook environment, I see the sites-only VCF and its index. Okay, so we're going to start with using Python to extract our variants of interest. And what variants are we interested in seeing? So there was a study that reported that deletions in the gene PDZK1 were observed to be associated with increased urate levels in the UK Biobank, which has implications for kidney function. I have included the reference to that study in the references section at the end of the notebook. So let's see if there are any deletions affecting PDZK1 in the all of us data. And we're interested in ones that are predicted to cause loss of function of this gene and that have an allele count greater than 40. So let's get set up. We're going to use PySam to parse the VCF in Python. So we need to import it. We're defining the minimum allele count to be 40 for the purposes of this demonstration. For right now, we're interested in deletions, but you could add other SV types to this list if you're interested in those. For right now, we're interested in SVs that are predicted to cause loss of function. So we put predicted LOF in this consequences list, but you can see the other functional consequences and add to this list based on the SV annotate documentation, which defines all of the functional consequence categories that we use. And we are interested in the gene PDZK1 for this demo. So you can add as many genes as you want to this gene set. But if you have a really long gene list, you probably don't want to be typing all of those out. So instead, if you have that gene list in a file, there's a code snippet right here to read the gene list from the file, assuming that there's one gene per line. Okay, so we've defined our variables. One last cell to define some file paths. So this is the input VCF, which is the sites only VCF. And we are going to extract these variants into a subsetted VCF, as well as a table with some key information. So this defines the input and output file paths. And this code snippet reads in the input VCF and searches for our variants of interest based on the variables that we defined. So that's deletions in PDZK1 with an allele count greater than 40 that are predicted to cause loss of function in PDZK1. So this code cell will iterate through the VCF and look for any variants that match those criteria and write them out both to the output VCF and the output table. So let's take a look at those outputs. And the way that I did that was just to cat the output table. And it turns out that there's only one variant matching all of those criteria in the all of us data. And it's a 194 KB deletion with an allele frequency of about 0.5%. And I've added a link to the UCSC genome browser for these coordinates that are encompassed by the deletion. So let's take a look at that. Okay, so I have the main select transcripts showing up top here, and we can see that PDZK1 is entirely deleted by this deletion, along with this other gene, GPR89A. So if it's fully deleted, we can see why it was annotated as being predicted to cause loss of function of PDZK1. Okay, so we've extracted some of the key information into this table, but there is a lot more information available in the VCF. So I'm going to print out that VCF record. So what this code cell does is it prints out the VCF without the header, and then it does some reformatting to put each of the fields of the VCF onto its own line. It also masks some of the fields to make sure that we're not displaying any participant counts that are less than 20. And I can run this command safely because I know from the table that there's only one record in this VCF. But if there are more records in this VCF, you would get a really, really long output. So just be careful if you're running this notebook and you change any of the parameters. Okay, so here is what our reformatted VCF record looks like. So even though I masked some of the annotations, there's still a lot of information here. So what can we learn from this VCF record that wasn't in the table? Some of the basic information was in the table, it's the same thing. One thing we can learn is that this variant has a pass filter status and a qual score of 80. It was originally called by read depth-based algorithms, so that's GTK GCNV or CNMOPS, as well as MANTA. It's supported by PE, RD, and SR evidence. It's predicted to cause loss of function in the other genes that we saw in UCSC as well, that GPR89A gene, and it also overlaps the promoter of another gene, CD160. And then there are also, let's see, one more thing I wanted to mention is that you can see that there are the NHOM alt field is 0. So that means that there are no individuals in the all of us data set who are homozygous for this deletion, homozygous alternate. And then there are a lot more annotations in the VCF that are related to allele frequencies for subgroups of participants, particularly by genetic ancestry group. But those are not displayed in this particular printout because some of those participant counts are a little bit small. But that information is available in the VCF and you can access it on your own if you have access to this data. Okay, So we've seen how to do this in Python. Let's also demonstrate how to do it with BCF tools. BCF tools is super flexible, it's fast, it's very quick to put together a command. So it's a really useful tool that we use really often in our group. So we can make the same table of key information about deletions predicted to cause loss of function in PDZ-K1 with allele count greater than 40 with the BCF tools query command. The -I argument allows us to specify what type of variance we want to include. So our criteria are deletions, predicted loss of function in PDZ-K1, and allele count greater than 40. And then the -F argument allows us to specify the format of the output table. So we're going to include these key fields: chromosome, position, and the variant ID, SV type, SV length, allele frequency or AF, and the genes in which this variant is predicted to cause loss of function. And we pass in our sites-only VCF that we defined the path for above. And then we produce this table, which I'm printing out here, which has basically the same information as the table that we created in Python, although it doesn't have a header because we didn't define that. We can also use BCF tools to extract the same subset of the BCF that we did before with Python using the BCF tools view command. And again, the -I argument allows us to specify which variants to include, and this is exactly the same as in the query command above. And this allows us to create a subsetted BCF. BCF tools does also work with a list of genes in a file, but we need BCF tools version 1.19 or higher. And we're in the process of requesting that the BCF tools version in Researcher Workbench be upgraded. But for now, we've just included an example of the command that you would use if you had that version. Okay, so we've looked at a lot of deletions today. And that is great because they're a really important type of structural variant, and you can very clearly see the types of evidence that are supporting them. But I want to look at something a little bit more complicated. So let's look at an example of a complex rearrangement. So we abbreviate those complex SVs with the SV type CPX. And I've just chosen an example variant with this variant ID. So I'm just going to use grep on the BCF to pull out that variant and then reformat it in the same way that I did before. So this is the result that I get. So this is a complex SV located on chromosome 4. It has a pass filter status. And the SV length is about 210 kilobases. So this is a pretty large rearrangement. And it's predicted to cause a whole gene duplication or copy gain of the gene UGT2B10, as well as a whole gene inversion or spanning inversion of the gene UGT2A3. And the allele frequency is about 0.8%. So those are all things that we are familiar with looking at from our deletion examples before, but this is a complex rearrangement. So to kind of dig deeper and really understand the structure of this rearrangement, the key fields that we're going to look for in the info column are CPX type and CPX intervals. So the CPX type gives us the subtype of complex rearrangement, and this is dup-inv-del. So this involves a duplication, an inversion, and a deletion all in one event. And the CPX intervals define the actual genomic coordinates that are affected by each of those types of rearrangements. So if we take a closer look at the CPX intervals, we can see that there is a segment of about 209 kb that is inverted. And within that segment, part of it is both duplicated and inverted, about 39 kb. And then there's a small segment about 1 kb that is deleted at the end. And we can view the entire affected region in the UCSC genome browser. And we can see that those 2 genes that were predicted to have consequence do indeed show up here in this region. So we can see where the information about the structure of this rearrangement is stored in the VCF, but it can be a little bit hard to wrap your head around just by looking at those strings of coordinates. So to better understand the structure of each subtype of complex rearrangement that we resolve with GATK-SV, there's a really good figure in figure 2 of the Nomad SV paper. So again, you can go to the references section at the end of the notebook to see the citation for that paper and go find that figure. For this particular example, it has the subtype dup-inf-del. So I've made a diagram of what is the structure for a dup-inf-del. So you can see that in the reference, we have these 4 segments that are labeled A, B, C, and D. And when we have this type of rearrangement, segment A is duplicated and it is inverted along with segment B. Segment C is deleted, so this shows the deletion breakpoint. And then segment D is unaffected. It's just there to show that the genome continues on after this. So I'll pause there for just a couple of seconds to let everyone digest this a little bit. So again, we have a segment A has been duplicated and inverted. Segment B has been inverted. Segment C has been deleted. Okay, so this type of diagram is really helpful for me at least, but it is still a toy diagram. So I also wanted to put it in context of this example rearrangement in the actual genomic region that we were seeing in UCSC. So when we look at UCSC and we actually take a look at the coordinates of each of those intervals that are affected by this rearrangement, we can create this diagram where we see both of our genes, UGT2B10 and UGT2A3, and we can see that when this rearrangement occurs, UGT2B10 is duplicated and its copy is inverted, and UGT2A3 is inverted. And then there's a portion of non-coding sequence after these genes that gets deleted. And so based on reconstructing the structure of this complex rearrangement, we can really see why the functional consequence annotations predict that there's a copy gain of UGT2B10 over here and over here, and whole gene inversion or predicted inversion spanning for UGT2A3. Okay, and at the end there are those references that I was mentioning. And that's the end of our demo, but again, this is available in a featured workspace, so you can come back to it at any time. And I want to jump back to the slides very briefly to acknowledge the work of so many people that went into generating this structural variant data. Many people from the Tolkowski lab and the Broad structural variation team. We also obviously couldn't have done this without the All of Us Data and Research Center, and particularly without the participation of the program participants. And I also want to give a shout out to Genevieve, who helped me prepare for this presentation.